I'm ready. Yeah.
Good evening. Good evening. Welcome to the Shoals Marine Laboratory's weekly science seminar. I'm Sarah Morris, the John M. Kingsbury, the Executive Director of the Shoals Marine Lab. For those of you that were here a couple of days ago, you should have known the name of the first director of the Shoals Marine Lab, John M. Kingsbury. We had trivia for those of you online. Thank you for joining us this evening for our seminar, the fourth Rock Talk of the 2026 season. For those of you joining us online who may be new to our community, the Shoals Marine Lab is the largest and oldest marine laboratory in the country that is focused on undergraduate training, both in coursework and research, and the lab is operated jointly by the University of New Hampshire and Cornell University. Our seminars are affectionately called Rock Talks because in the early days of the lab, before we had walls and working ceilings, our seminars were actually literally on the rocks, and we retain that name as part of our history. We've migrated indoors, but these weekly seminars provide an opportunity to bring the entire community together to hear about different aspects of research related to the marine sciences and the environment. In addition to our guests joining us remotely this evening, we have 3 classes with us on the island at this time: marine mammal biology, marine invasive species, and sustainable fisheries. In addition to the students taking those courses, we also are in the second week of our 10-week surge program, our research interns. We're excited that everyone is here and excited to be here this evening. And we're glad to welcome our friends who are joining us this evening online. The format for this evening's seminar is 45 minutes of the talk, followed by 15 minutes of Q&A. Those of you that are online, please use the Q&A button at the bottom of your Zoom screen. Those of you in the room, y'all, I think you're used to me now, where we'll be coming around with the microphone, making me run from various sides of the comments so that everyone can hear you both in the comments and online. This evening's speaker is Nathan Furey. For those of you who were expecting Ashley Busheko, this is not Ashley. Ashley had to cancel because of a family situation, and she will be rescheduling for some point in the future. We were thrilled that Nathan was available and willing to come join us this evening as our fourth Rock Talker. Dr. Nathan Furey is an associate professor in the Department of Biological Sciences at the University of New Hampshire and leads the Fish and Movement Ecology Lab. He received his bachelor's degree in marine biology and environmental science from the University of British Columbia, his master's degree in wildlife and fisheries science from Texas A&M University, and his PhD in forestry from the University of British Columbia. Nathan's research attempts to quantify the motivations and consequences of animal movements across aquatic taxa and freshwater, estuarine, and marine ecosystems. Many movements are in response to foraging and avoiding predation, and thus the Fish and Movement Ecology Lab also conducts investigations in predator-prey interactions, diet, and bioenergetics. This seminar will explore multiple model systems where predator movements are uniquely influenced by prey availability with potential consequences to fitness. These systems span salmon-bearing systems in British Columbia to the Canadian High Arctic to the Gulf of Maine. Dr. Furey was HH's Outstanding Assistant Professor of 2024 and is the inaugural recipient of the Robert Summers Professorship of Biology at UNH. This evening, he will be presenting Following the Food, Variation in Trophic Resources Drives Aquatic Animal Movements. Please join me in welcoming Dr. Nathan Furey.
Awesome. Well, good evening, everyone. Thank you all for being here, even if you're kind of forced to be. I'm very excited to be here and be able to share with you some of the research that I've done over the past, honestly, over a decade, but also with the contributions in my lab. Before I begin, there's a lot of folks to thank, a lot of organizations that help make this work happen from a funding standpoint, but also a logistics standpoint. I also really want to highlight the Honeygatine First Nation, who gave us access to the incredible field site that we're going to start with this evening. And certainly I'm the one up here who's getting the glory of speaking with you all and sharing all this cool science with you, but it really takes a team. So I'm really reflecting the work of my lab, of our undergraduate, graduate students, postdocs, and staff as well. And so I am known kind of in a colloquial way on campus as the fish guy. I am a fisheries biologist by training, but I also consider myself a movement ecologist. And so what does that mean? That means a lot of my lab tries to understand why do animals move and what are the consequences of those movements. You don't need a PhD to think about, well, why does someone or something eat? To kind of think about, well, why does something move? It's to eat or to keep itself from being eaten. So my lab also does research in predator-prey interactions, trying to identify what influences those predator-prey interactions. And we all eat, or when we move, those are all actions that either to gain energy or to spend energy. So we also do research in the field of bioenergetics. And whether we're talking about movements in space use or how or what you're eating or what is the energy gained or lost from that, we need a perspective of where in the world are you. So my lab also tries to take a landscape ecology lens in our research as well to understand how differences in habitat, latitude, or ecosystem might affect all of these ecological processes. And so as I tried to think about a seminar that would maybe encompass all of these themes and give you a real sense of what my research is about, I kind of came on this question as a unifying theme for tonight. How do variations in prey affect predators in terms of their behavior, their movements, their feeding, and ultimately their fitness? So as Sarah's introduction alluded to, we're going to kind of have 3 stories or vignettes tonight. The first where I did my PhD in postdoctoral work in British Columbia. Then we're going to move to the high Canadian Arctic. And then we'll end up very close to your all's home this summer, the Isles of Shoals and the Gulf. One of the commonalities across all of these projects is the technology that we use to assess animal movements. And that's using what we call telemetry. And so when we're tracking critters in the aquatic environments, whether it's freshwater, estuarine, or marine environments, we're using acoustic telemetry. So this is the idea that we have an electronic tag that we can surgically implant or attach into an animal. And then we can have receivers or listening stations or hydrophones placed within that environment in the water. And these tags send out little sound signals at frequencies that we can't hear, but that these devices that we place in the water can hear. And so if that tag happens to be swimming by close enough to that receiver within proximity of it, that receiver will log that this fish or this tag was here at this place and this time. And this gives us really rich individual level information on the movements of animals across habitats. Now, if your critter is not in the water, this can actually be quite a bit simpler. So for our third vignette tonight, when we talk about the movements of common terns, we're using GPS or satellite tags that obtain information from space and then offload their information via radio signal, very similar to what's in the product. So let's dive into our first story that is going to be focused on maybe a character not all of you are familiar with, but certainly Mike Sigler is, and that's the sockeye salmon. Sockeye salmon is maybe the most iconic animal migrant on the continent and certainly among fish. They're found throughout Western North America, and their spawning migrations are absolutely a spectacle. You have literally millions of fish that are returning from the ocean, moving upriver, sometimes thousands of miles to go back to the same river they were born in and to spawn. And even if you don't know a lot about sockeye salmon, I'm guessing several of you grew up watching nature shows where you saw, maybe you didn't care so much about the salmon, but you saw all of those incredible charismatic megafauna coming in to feed on those salmon. How many of you have heard of Fat Bear Week? Okay, right? So that idea that you've got this huge pulse of migratory salmon moving through a landscape, and you've got a predator that's responding to it. So these migrations can feed ecosystems and feed predators that are part of those ecosystems. Now, while the adults are what get all of the press, let's say, and for good reason, they represent important commercial, recreational, and indigenous fisheries. There's actually another migration that occurs during the sockeye salmon life cycle. and that is by juvenile sockeye salmon smolts. These are juvenile salmon that make a physiological decision, if you will. Apologize for anthropomorphizing, but they undergo a physiological transition to be ready for saltwater, and they move downriver and out to the ocean. And so this also represents a very large migration, long distance migration, that could also act as a large energy source for predators moving across landscapes. And so I was very fortunate that I did my PhD and postdoctoral work at Chilko Lake in British Columbia, which is in the Caribou region and in the Huntington First Nation territory. And it is home to one of the largest populations of sockeye salmon in British Columbia. And because of that, the Federal Fisheries Agency, known as Fisheries and Oceans Canada, kind of akin to the National Marine Fisheries Service in Canada, install this river-wide counting fence every spring to enumerate or count how many juvenile salmon leave that lake and start their migration downstream. That's a really great platform for me as a researcher trying to get my hands on migrating fish. Now, I mentioned that this is a really large population, so I want to give you a little bit of a sense of that. So we're going to dip our heads underwater. We're looking downstream through that counting fence. And over a 4 to 5 week period between mid-April to mid-May, between 10 and 70 million of these juvenile salmon will leave that lake and head downriver. So this indeed represents a very large energy pulse leaving that lake and heading downriver and out to the ocean. And so I'm not going to spend a lot of time on my PhD, but a lot of my PhD was using acoustic telemetry on these little fish and trying to understand what are the factors that affect their survival as they go downstream. But one of the consistent findings was that survival was really poor early on in the migration in the Chilko River. So this beautiful image by a former student, Steve Healy here, shows Chilko Lake on your right, outletting into the Chilko River on your left. And we found that year after year, the first 13 kilometers or so of this migration was really high risk. In fact, we could take these baby salmon, put them into trucks, truck them downstream of this spot, and they experienced higher survival. In addition, we saw that the fish were really only traveling at night. So if you have poor survival and fish that are only willing to move at night, those were pretty good indicators to us that maybe predation is important in this landscape. So I was able to convince my PhD supervisor, like, hey, let's not just work on sockeye salmon. Let's start investigating these guys too. These are bull trout. So bull trout are a relatively large, long-lived member of the genus Salvelinus, or the char. So this is very closely related to like lake trout and brook trout that we have here on the East Coast. And we would notice that me as an intrepid PhD student getting to my field site a couple of weeks before the juvenile salmon were migrating, prepping all my gear and everything, we would look in the river and we would see maybe a few hundred of these bull trout sitting in the river as well. Wow, that's interesting. You don't see these fish aggregate too often normally. Like, what are they here for? What could it be? So we started to just see, do bull trout eat sockeye salmon smolts? And if so, how many? And the answer to this is yes, they do, and quite a few. So these two images here are stomach contents from individual bull trout. So the one on the left is stomach contents from one bull trout. The one on the right is contents from another bull trout. The one on the left has 65 sockeye salmon smolts in it. We had some stomachs that were upwards of 100 sockeye salmon smolts. That one on the left, if I didn't tell you it came out of a stomach, you'd probably believe me if I told you I netted it out of the river, right? So it really implies that those fish were eaten over a very rapid and short time period and very close when we sampled that bull trout. That's a little morbid, but in fact, when we dumped those fish into a cooler, some of them tried to start swimming around again. So these bull trout are absolutely feeding at high rates. If you still don't believe me, let me show you in one other way. So this is a stomach that was digested out of a bull trout donated to us by the Huntington First Nation. And hopefully you can see a bunch of sockeye salmon looking back at you. If you can't, let me help you out. Okay. And so we're not going to go through kind of the mathematics behind it, but we use things called bioenergetics models to quantify just how much are these animals feeding. And these are instances that we would call hyperphagia or binge feeding, whereby these animals are eating way more, upwards of 10 to 30 times more than what we would expect them to be able to eat and digest in 24 hours. So I know I can eat a lot during certain holiday seasons, but I can't match the bull trout. And so that was some of the work I did during my PhD. I was then fortunate enough to get a grant along with my supervisor, Scott Hinch, to fund another student to continue these investigations between bull trout and sockeye salmon. So while I was focused on this spring migration, there was still another pulse that sockeye salmon could provide to bull trout. And so Adam Hannigan also went back in the fall and pumped stomachs from bull trout to see what they were eating then. Now, the fall is when the adults are coming up and spawning, those initial migrations. There's Adam. He's wonderful. He just started his new job last week. So I'm a little sad about it. But very excited for him. Just finished his postdoc with us. This is another image of the stomach contents of an individual bull trout, and those are all sockeye salmon eggs. So we also found that these bull trout were exploiting this seasonally distinct pulse, not only in the spring, but also in the fall. In addition to eggs, they would also sometimes feed on sockeye salmon carcasses, because sockeye salmon are what we call semiparrots. They only spawn once and then they die. I like to think of them as the most romantic animals on the planet. I'll talk more about that later if you ask me to. And so I'm going to show you a few figures that helps quantify some of these impacts that feeding on these pulses have that Adam published last year. So I mentioned myself, I really like food. You probably all saw me seconds of lasagna because it was so delicious. You put me on an ice cream bar, I will eat for a half hour straight. I love ice cream, I love sweets. But I do not have the ability to change my gut size. And you're thinking, well, duh, right? Well, what we're finding more and more is that fish are pretty darn plastic. And what I mean by that is flexible in their phenotypes or flexible in terms of their traits. And so what Adam also did is he went and dissected, we got the permission from the Honey-Gatine First Nation to actually destructively sample some fish and looked at the sizes of their digestive tracts and other parts that are important to their physiology and to their digestive machinery across the seasons. And what we found was pretty darn remarkable, but is being seen in other systems dominated by salmon resource pulses, that they actually go through seasonal shifts in their digestive machinery. So things like their intestine and their stomach size actually are larger during the spring and in the fall when these resource pulses are available. And then in the summer, when things are more energetically costly because waters are warmer and food is a lot harder to find, these things contract because it takes energy to maintain digestive machine. So we get this incredible seasonal changes in the physiology of these animals, and it has an impact on the condition of these animals as well. So if you focus on the left and that left-hand axis, a condition factor is just a fancy word fish biologists use for a modified weight to length regression. How thick are you for your given length, essentially? How much mass do you have for your given length? So the higher the number, the fatter you are for your given length. And so we see that these animals are in higher condition during the seasons that the sockeye salmon are there. Furthermore, though, it matters where you are in this lake. This lake is actually pretty darn long. It's almost 70 kilometers long. And you need to be on that right-hand side of the plot. So that dotted line and everything to the right means you're at that outlet where the lake turns into river, where things are spatially constricted, where it's easier to feed. And if you're there, when the salmon are there, you are going to be in higher condition than if you are elsewhere in the lake or during other times. So hopefully this helps cement that sockeye salmon is important to bull trout. But I'm a movement ecologist. I wanted to know, well, what are the bull trout doing about it? What are their movements in response to these migrations? And so we're going to look at this at two different spatial scales. And we're going to start with a fine scale study where we're going to call this area the gauntlet. This is about a 1.3 kilometer stretch that really represents the transition from the lake environment to the river. So the sockeye salmon smolts are leaving the top of the image and going down towards the bottom of the image, and eventually through that counting fence at the bottom of the red polygon. And so with this, we used kind of a unique form of acoustic telemetry. We put something like 45 acoustic telemetry receivers just in this 1.3 kilometer stretch of the river. And what that allows us to do is actually estimate positions of animals rather than just their presence. So usually when you have an acoustic telemetry receiver and a fish gets close enough to it with a tag, you know, okay, you're near there. But now with this system, we expect that 3 or more receivers actually get the same signal from an individual transmission from a tag. And we can actually trilaterate that down to a few square meters. So now we get fine scale positioning of these structures. So this is a wonderful animation put together by Adam that represents 48 hours. There we go. 48 hours of movement bull trout. So each dot is a bull trout. And they're just going to be color coded based upon the time of the day. So yellow day, blue night. And it's kind of boring during the day. So they don't move around a whole lot. They find 3 or 4 spots. We call them hidey holes, deep little spots for them to hide during the day. But then at night, which happens right now, you see the movement rates increase dramatically and they went up to the fence. And then as soon as it was daylight, they went back to their deep little hidey holes. Then we're going to get night again. The increase in the rapid movements, they push up towards the fence where things are shallow. And it's easy to sockeye salmon cells. And this bears out in the data. This is a manuscript that we just submitted the revisions for. But the bull trout move at faster rates and they use larger areas at night relative to the day. And they're positioned much closer to the fence at night than they are during the day. So these bull trout are indeed making behavioral responses to the availability of the sockeye salmon migrations that occur each spring. But this is probably pretty darn intuitive, right? Like, no offense, someone carts in some ice cream here. I'm going to start to ignore you all and head to that part. If food comes by you, you're going to respond to it, right? You all were here for dinner. The bell went. You all got up and moved towards the plates, right? We were curious as to whether bull trout were willing to move farther distances to be in the right place at the right time, a position to make those final scale. So then we expanded our investigations much broader, where we put acoustic telemetry receivers throughout this 65, 70 kilometer long lake, as well as in some nearby river systems. Now, I'll admit I was working on a new figure for this, but didn't have time. This is incredibly cherry picked. It's 2 fish, but I'll give some context a little bit. But look at the movements of this fish. They use the entire lake, but now in about April, right now, in May, when the smolts are there, they run up. During the other times, they use the entirety of the lake. They come back up in October when the sock adults are spawning, and then move around a little bit more. And then back in April again, they're going to come back up. And in fact, between 30 and 35% of our tagged bull trout moved more than 30 kilometers in either October or April to be in the right place at the right time. But it was incredibly variable. Some were there for both. Some were there for only one of them. Some of them went for multiple years in a row. Others would kind of skip. And this makes sense, particularly in the fall, because that's also when bull trout spawn. So it might be make a decision. Do I spawn or do I go binge myself on some sockeye salmon? But regardless, it seems that some of these bull trout are indeed moving long distances in response to migrant salmon. So we have predators that are migrating long distances in response to migrating prey. And I just thought this was the coolest kind of phenomenon. We've got energy pulses moving across an environment, and then we're starting to have other things chase it or attract it. And so we coined this as a new term called migratory coupling, whereby predators are willing to move beyond what we call their home range to go track or intercept migrant prey. Now, if you're willing to do this, you're willing to go move tens of kilometers across this lake to go be in the right place at the right time, there better be a benefit to it. And so one of the other things that Adam did, and this is another paper that's in review, is he took non-lethal fin ray biopsies. So he took a little piece of bone from each of the fish that he either took their stomach contents from or he tagged, and he used that to age them. And I'm happy to talk about how we do that as well. But you might be talking about it in your fisheries biology class. And so this is how old all of the bull trout that Adam touched were relative to their size. And then we can put a simple model through that. And then all of the colored ones are the ones that we tracked their movements with acoustic telemetry. And so you can then see fish that fall above that line are therefore big for their age. I was not, in terms of height, I was never one of those kids. If you fall below the line, you were shorter for your age. And if we pull those out between those that exhibited the migratory coupling at least once, so the one or the greater than one, versus those that never did those large scale movements to be at the right place at the right time. It seems that the bull trout that undergo migratory coupling are bigger for their age. So there's a growth advantage there. It's always good to be bigger as a fish. That means fewer things can eat you, and if you're a female, you can generate more eggs. So that kind of wraps up vignette number 1. But then in 2018, I got a job at a place called University of New Hampshire. And it just led me, thank you, Sarah. It got me thinking, well, where can I study similar things? Where might these trophic hotspots be even hotter, possibly? And I was very fortunate. I was still living in British Columbia, figuring out my move across the continent. And I got a call from a good friend and colleague of mine, Nigel Hussey. And he has a wonderful British accent that I wish I could impersonate, but I can't. So Nigel called me and he said, hey, Nathan, I hear you got a professor job. Do you know what you're doing? No, not at all, Nigel. He goes, I got a wonderful project. We have way too much money and way too much data and samples, but not enough people. Do you think you could recruit a graduate student to go there? And I said, I think I could do that. And so I got to give Lars Hammer a lot of credit, because even before I met Lars in person, I was literally in British Columbia still. I was sending him on a plane to go work with Nigel. And I feel pretty fortunate to work in British Columbia, but this is where I sent Lars to. And a year later, this is where I sent Nate Herman to. So these were 2 master's students that I sent up to the High Canadian Arctic to work with Nigel Hussey, Fisheries and Oceans Canada, World Wildlife Federation, a bunch of other NGOs and such. And this place is known as Tremblay Sound in Nunavut. It's a coastal sound, so it's a marine environment. Very small. You can see shore to shore, even in that photo, right? But it is very well known by the Inuit for its productivity, particularly in spring. And so like a lot of Arctic coastal systems, you go through periods of very long, cold, dark winters where you have ice on, which is what you're seeing now. And then in the spring, which is really like June, that ice comes off and you get a big boom of productivity. And this is because the days are getting longer much more rapidly. And so obviously primary productivity can increase, but there's actually a jump start because that ice has critters living in it. You have what we call sympagic or ice associated critters, including things like amphipods and isopods and such. So when that breaks up, even before the primary productivity really ramps up to generate secondary productivity, as we see in most ecosystems, you actually get a little pulse of secondary productivity right away. Vertebrates. That's just a little cute little sympathetic. And so this sound is really well known for how stark its spring pulses of energy are, such that the local Inuit harvest narwhal. And you have over 1,000 narwhal that come into this single sound every single spring. Now, this is the I'm not talking to you about narwhal tonight. I'm sorry. And that's because those are the type of charismatic megafauna that Nigel Hussey gets to work with. And I encourage you all to look him up. It's fantastic. He said, Nathan, I'm going to work on the belugas and the narwhal and the Greenland sharks. How do you feel about working on sculpin and Arctic char? I said, that's my jam. And because all fish are beautiful. All right. And so these are 2 really cool contrasting kind of taxa as well, because you've got Arctic char on the bottom there that are migratory. They're anadromous. So they're going to migrate between freshwater and saltwater to complete their life cycle, while sculpin are not. They're quite lazy. Like they like to stay in one spot. And so we had similar questions. How do these 2 different taxa with different mobilities respond to this incredible pulse of productivity in spring? And these were the theses, master theses for both Lars Hammer and Nate. So that image of a sculpin on the top left, that's what I like to say is an average sculpin. This is a sculpin during the ice off. So that is a softball with fins, right? And so just absolutely stuffed with amphipods. This is a stomach dissected from an Arctic char. And you can just see it is stuffed full of amphipods. And so in terms of what does this actually look like, here's some data from a paper they just had published last year. They were joint first authors on this, comparing the feeding of these 2 species. On the y-axis here, we have a metric called relative consumption. That just means what percentage of your body mass was your stomach contents. So you're seeing here, you know, anywhere from 1, but all the way up over 5%. So they actually weren't binge feeding quite as heavily as the bull trout at Chilco, which is pretty darn incredible. But this would be me like having 8-ish pounds of food in my belly. And I did love the lasagna tonight. I did not eat 8 pounds of it. So they're obviously feeding at very high rates too. But what does it mean for their movements and how they respond? And so this is a little animation that Lars put together. And what he found was that a handful of his fish actually overwintered in a river habitat that directly connected to the south. Can't see it on the map, so I used a little red arrow there to show it. And for those fish that overwintered in that river, they actually, and it's going now, they distributed themselves throughout the sound even before the ice melted. It's as if they were saying, hey, we know the food and buffet is coming. Let's spread out and let's all be in position to be there when it all falls in place. And this is actually quite unique in this system because most riverine systems in the Arctic are actually too shallow for this to occur. The ice would actually block movements or migrations until the ice opened up. So this might be a little clue as to not only why is it such a hot spot for things like narwhal, but this is also a really popular area for Inuit and other First Nations to harvest Arctic char. And the movements of these Arctic char, whether they came from that river or from other systems further away, were directly tied to that ice off. Such that here you're seeing those Arctic char and how many of those tagged fish we actually saw in the system. And we left these receivers in year round. So if they were there other times of the year, we would know it. But you'll just see zeros across the board. None of the fish we tagged were there until you see those two big pulses or peaks. If I overlay the ice coverage, Lars actually took satellite imagery of the ice to estimate the ice coverage every week. You'll see how tightly linked it is, but that a bunch of fish get there in that week before the ice actually melts off. And then they don't stay long. So it's almost like once that ice has dissipated and that pulse of sympagic critters have fallen through, they just go on and move somewhere else. So it's not just about a warm season, it's connections to those ice. But what about the sculpin? Sure, they eat a lot, but they're little sculpin. Do they move in response to this? And so this is a wonderful little video put together by Nate, where you're going to see the ice imagery on the right, but then you're also going to see our acoustic telemetry array and then the movements of these sculpin. And it's going to be color-coded when the ice is off or melting, it's going to be yellow, and when it's winter, it's going to be blue. And what you're going to see, so right now, you actually see when it's yellow, the fish are bouncing between those letters. They're actually doing relatively large-scale movements. But when it's blue, they do those much less often. And so what we see is that even this sculpin that we don't think is very mobile is increasing its movements. It's worth it to increase your movements when that ice is off and your food goes available. So pretty cool kind of comparison and contrast to those. But I do recognize we're here at the Isles of Shoals. I do recognize that we're by the Gulf of Maine. So why don't we end tonight with third story, where again, the first two stories were like, it's just so obvious, right? Like maybe no one has gone and recorded data of it, but certainly our Indigenous partners and Indigenous communities knew a lot of these phenomena were already occurring. But what about in a system where the food quality and food abundance and its variations might be a lot more nuanced or variable season to season or year to year? Could those also affect the movements of animals? And I am team fish, but I am also learning to love team bird too. So we're going to talk about common terms as well. And this is only possible through the incredible efforts of Dr. Elizabeth Craig, as well as Alia Caldwell, a PhD candidate in the lab. And so Dr. Craig has been leading efforts for a long time, and people before her as well, to monitor the common terns at White and Seavey Islands. So I've just in that little red box there, you can kind of see where the Isles of Shoals are, where we all are sitting right now. And so Liz's team collects an incredible amount of high quality data. But one of the things that we've been doing since I've come to UNH with Liz is using GPS or satellite telemetry on these birds to look at their movement. So this is an animation that Alia put together. from just 1 year of data. And so each little line there or piece of spaghetti is a different bird. And what you'll notice is that the birds change colors. And this is because we use a statistical model known as a hidden Markov model to actually try to guess when that bird is flying around, what is its behavior? And the blue is when we think they're foraging. You might be thinking, well, Nathan, that kind of sounds like voodoo, black magic. How can the model do that? It's actually very simple. All we're doing is looking at how fast is that bird moving and how tortuous or how much turns are they doing. So let's use me, man. I keep kicking this thing. Let's use me and my love of sweets as another example here. So if you were to put a tracker on me in a grocery store, okay? Now you've got the produce aisle. I do love greens. Let's just take this with a bit of humor. But let's pretend I just walk on because I don't care about the produce, right? I would not have many turns because I'm just trying to get through, and I'd be walking more quickly because I'm not looking at anything because I just know I don't want it. I'm not where I want to be yet. Then I get to the ice cream aisle, and it's like, ooh, Ben and Jerry's over here. Ooh, what is this frozen yogurt thing? That looks good, right? My movement rate goes down, and all of a sudden I start turning more. All a hidden Markov model is doing is trying to separate those out. When does the animal slow down and turn more versus when is it just moving? And so if I play this video again, hopefully you'll actually kind of be able to see that as you go. And so everywhere blue is where the animal is foraging. And we see some foraging hotspots start to emerge. And I wish I had a pointer. I don't think, can I show you? No, I can't. But one of them is Great Bay River mouth, the Piscataqua River, Great Bay itself. And then just to the south of that, you see another hotspot. That's the Merrimack River mouth. So these river mouths are really important. And then you see just north of Cape Cod, not quite as tight of a hotspot, but still a bunch of foraging locations. That's Stellwagen Bank National Marine Sanctuary. So river mouths and then this sandy bank, that's a great spot for critters like sandlance, tend to be where these birds forage. And so what's really cool is not only that through Alio and Liz, we're able to track the movements of these birds, but then we can integrate that with other information that Liz collects. Like, what are these terns feeding on? So through surveys through blinds, as well as through GoPros, we can actually see what the adults are bringing back to their chicks, what they're provisioning. This is an example of a herring species being brought back to a chick. Liz's team also looks at how productive is the colony, how many chicks do they get. And so one of Alia's PhD chapters, which she just submitted revisions to this past week, if you're listening, Alia, nice job, that I will share with you all now. So let's start with just how did the colony do across these 4 years that we integrated these data together. In 2022 and 2024, the colony did pretty well. On average, you had about a chick per nest make it and fledge and survive. This is in contrast to 2021 and 2023, when we had less than half a chick per nest survive. And so through starvation or other means, we had a lot of dead chicks, and dead chicks are sad. If we then link that to the prey provisioning information, Alia kind of categorized the different critters that were being brought back to the nest and kind of said, yep, that's something a tern would really like to eat and has a lot of energy versus less. And if we look at kind of the abundance of the high quality prey items in 2022 and 2024, more higher quality prey items were being brought back to the nest versus in 2021 and 2023 now. Now, this doesn't mean that these necessarily were all bad prey in terms of their energy density. That's another thing our lab does, is look at how many calories are in different types of prey. But it might just not be the right thing for what that tern eats. So for example, on the right here is an image of a butterfish. Butterfish are a perfectly wonderful fish. Again, all fishes are beautiful, but they're just very deep bodied and cylindrical, such that a tern chick can't actually swallow it. So oftentimes they get back to the nest, the parent tries to give it to the chick, and then the chick says, no, I can't physically eat that. And so we put these two together to kind of say, hey, we kind of have 2 years that things were pretty good and 2 years where things were not so good from a productivity standpoint and what they're eating standpoint. So then kind of Alia's next question was, well, what does this mean in terms of how they're moving around and trying to find those prey? And so now you're seeing the movements for an individual year, kind of across the entire colony. And those polygons just kind of represent minimum areas that constitute the area's use. That broader one, essentially 95% of all of the foraging points fell within that broader polygon. And what we see in 2021 is that that's a really big polygon, and that it spans all the way from New Hampshire and Great Bay, all the way down through Stellwagen Bank National Marine Sanctuary, and all the way around Cape Cod. Versus in 2022, when things were better, you can see it much more contracted. There's still stuff around Cape Cod, but now you can see that separation where Stellwagen is not used in the same degree, and much more of it is focused around those immediate, closer by waters around the Isles of Shoals. And this is a pattern that we see consist in 2023 and 2024 as well. And if we put actual numbers to the sizes of these areas, we see indeed that these terns are moving further in the years that the prey quality is lower. So the parents are trying, but they're not necessarily able to compensate enough, right? Because we still see that the chicks aren't doing as well. So this is how we can start to see how movements become important, or at least affected by differences in prey availability and prey quality among years. Now, I'm going to end with a little bit of a tangent, but it's just so cool that I can't help share it with you. So the backpacks that Liz and Alia and the team uses to track these birds, in general, they fall off because after the breeding season and these animals migrate, they lose enough weight that the backpack can kind of slip off. And then we stop getting data. However, we had an instance where the team went back, where Liz's team went back, and they noticed one day, there's a bird already wearing. And it immediately started to offload all of the data it had been collecting over the prior year. Because it connects with satellites to get its position, but to give it to us, it's got to get to a radio station that we have here at YPCB Islands. So we think this is the highest resolution annual track of a common tern, kind of on this side of the Atlantic. Did you see how quickly it got down South America? And it is spending all of its winter off the Amazon River mouth, is that area that we're seeing there. And then what we're going to see here, we're approaching May. We're going to see how quickly it shoots right back up. It did spend a couple of days along the middle of our coast, but then popped right back up. And this might seem tangential to our story here, but when I see this, it's just like, oh my goodness, we are just seeing such a snapshot of when they're feeding and when it could influence the fitness. And there we're thinking more about the fitness of their chicks. But they are undergoing incredible migrations and then having a whole season of probably pretty intense feeding off the Amazon River. What things are they eating there? What's the quality of those prey? How does that vary year to year in space? It shows us just no matter how much work we do in a system, how we're really only scratching the surface on any of these ecological stories. And so with that, I'll just end with a few conclusions as you look at these beautiful sockeye salmons. And that's trophic resource variability. So kind of these hot spots or these not spots affect predator movements both across space. Predators are going to respond to the energy that's available to them. Variations in the quality of those prey that they're consuming are going to affect those predators. We saw that most clearly in the last story. But that there's a lot more to understand in terms of what is driving variability in response to predators. Why do only 30 to 40% of the bull trout in a given year seem to react to the sockeye salmon pulses? A lot more for us to investigate. And so with that, I will stop there, and I'm happy to take any questions.
Thank you so much, Nathan. Questions in the room?
Thank you so much for that talk. That was really, really engaging and awesome. I was curious, you had mentioned when you were talking about the sockeye salmon that you felt like they were the most romantic animals on the planet, and that you would talk more about it. And I'm super curious about that.
Yeah. So, I mean, imagine a better love story than this, right? You grow up and then you just get a call to leave home. And then you just go. You just go. You don't know what's there, and you just run downstream through canyons and fast-moving water. You have higher salinity. You've got seals and birds coming after you, and you just have a drive to then eat as much as you can. Be a glutton of life, if you will. And then all of a sudden, something calls you back home. 2 to 3 to 4 years later, Pacific species we're talking about. And you decide, you know what? I have one purpose in life, and that is to get back home and help the next generation. So you migrate thousands of miles, and you are doing it using so many senses, maybe magnetic reception that we're still trying to understand, maybe visual observations. But once you get close enough, you're using your nose. You can literally smell home. I'm sure some of you kind of know that scent, right? The scent of home. And then you just follow your nose till you get to that place that you were born. And then you go, you find a mate, you defend each other ruthlessly from others trying to steal your spot. You build a nest right in the river, and then you spawn, and then you spend your last dying breaths defending those eggs. And then eventually you succumb to this long journey and to the senescence. And then in your last final act, you decompose and you help fertilize the rivers that your babies will grow up in. And there is science behind every single one of those things.
Great talk. Thank you so much. My question was about one of the graphs from the first study.
Yeah. So
It's the one with the trout that went back to the gate 0, 1, or more than one times.
Yeah.
I remember the 1 and more than 1 being pretty similar, but it looks like maybe there was a trend of the ones that only went back to the gate once being larger. I was wondering what a possible reason for that might be.
absolutely. Sorry. Try this way. So sorry, your question is like, why did going back more than once not do better than just going back once?
Yeah.
Yeah, that's a fantastic question. I think there's a little bit, one, small sample size theater, a little bit. Two, I think there's a little bit to dive in there. Some of the ones that only went back once went for the fall spawning of eggs. And in theory, that should be a higher profit season to go because those eggs are just so energy dense. And there's very little of it of what we would call an indigestible fraction in terms of bones and things that are hard to digest. But again, it's a bit of a choice because bull trout should be spawning around the same time as well. So there might be a little bit of a link there, but I think we would just need a lot more sample size and do a lot more. Sure. But fantastic question. It's one that we're racking over.
That was really good. I was wondering if with the baby funds and other issues with the basic size of the fish, if we're already starting to see adaptations within the baby curves, or if this is more of like a cyclical process that happens over years.
Yeah, and so it does tend to be when it's warmer because butterfish tend to be a fish species that we associate more with the mid-Atlantic bite rather than the Gulf of Maine. So it tends to be when we have warmer summers that butterfish come further up and then seem to be more available. And then it's a bit of a double-edged sword because then when those species are coming up, that tends to also then be when our colder water species like herrings that they do want to eat, have really high energy density and are thin enough to eat, tend to get pushed further north and away as well. In terms of adaptations, I think that's probably a really hard one because these terns are what we call gate limited. So it's basically how far can you open your mouth? And it would be quite a large jump in that gate for them to be able to start swallowing.
Thank you so much. How rigorous of a process would you say was it to do research work outside of the country?
Yeah, so in terms of, so for the first vignette, I was living in Canada during that whole time. But obviously that still had a lot of logistics when it's a 10 to 12 hour ride and the last 2 hours are on a logging road and You have no cell service or anything like that. And a lot of it, both for British Columbia, but then also working in Nunavut, the Tremblay Sound, the second thing, was a lot of working with the indigenous communities, as well as those folks who had already been working in those areas for a long time. And having consultations and kind of direct conversations as to what we want to accomplish, but also what do they want out of the project if they are interested in the project, obviously permissions for the project. Now, for the Tremblay Sound, the Arctic work, I was in the US for most of that work when Lars and Nate were up there. And certainly there were some things that we had to figure out. So like what type of visa do they need and how long can they stay and all of that. And we also, a lot of the samples were housed at Nigel's lab at the University of Windsor. And so that took coordination as well to send Lars and Nate back to Canada to go do dissections and things like that as well. But I just have to say I was super, super fortunate to be working with an incredible team that we're all really excited to have us there. And that just made our lives easier. I like to say I am not an Arctic ecologist. I just have the incredible opportunity to be able to go dabble in that and help out in that. Because I myself have never been to the Arctic. It is a very rough place to live. I actually, usually say that, that field camp that you saw there, there's no facilities there, right? So you're living in tents, you're doing your number ones and your number twos in buckets. Usually I use some 4-letter words, but I know this will go on YouTube. So it's a very remote and harsh place to work. And people who have made their careers working Arctic systems are some of the most incredible, badass people that I know. I was just very fortunate to have a lot of those logistics figured out for me.
I really enjoyed your talk. Thank you.
Thank you.
I wanted to go back to Nunavut.
Yeah.
And I thought that data that you showed about the Arctic char and the ice breakup and the timing of how they left so quickly afterwards.
Yeah.
Could you speak to what the predators of Arctic char might be and if that had any bearing on them then leaving really fast?
Yeah, that's a incredible question. Certainly the narwhal could take them out. The Greenland sharks as well could be feeding on them. Although we do think both of those are more tightly coupled, probably to polar cod than to the Arctic char. So certainly predation could be an impact. That's kind of one hypothesis, and it's probably a mix, right? It's like, do I want to stay here? Like, what's the profit of here versus the risk of here? And so I think the profit side of it is we kind of almost imagine there's all these little sounds going up the coast. And so it's almost like, are they just riding the ice breaking off as much as they can? Ooh, let's dive into here. The ice is breaking up. Okay, now let's get out of here. Can we go find somewhere else the ice is breaking up? Because again, we saw the fish that came from that river got there beforehand. We would see fish that came from presumably other freshwater systems would come in oftentimes like right as the ice was breaking up. So in short, don't have a good answer for you, but a couple different hypotheses. But I like the predation result.
Thank you for the talk. I wanted to ask about how do you think climate change is going to affect the movement of prey and predators in the sound with, there's going to be different amounts of things with ice coverage.
I mean, this is one of these systems that we might lose ice completely in my lifetime. And so when we see both of these species where their dominant prey items are these sampagic or ice-associated critters, that's kind of worrisome, right? And so we definitely expect this to affect the migrations of these animals and also what they're feeding on. Now, it's not to say there isn't other stuff to feed on. There is. It just means they're probably going to have to exhibit quite a bit of plasticity or flexibility in terms of how they respond to those. Because like in theory, well, if you have ice off for longer, then you just get more of that primary productivity, more of that secondary productivity. And we're already seeing that in certain fringe areas of the Arctic. We call it the Atlantification, where it's starting to act more and more like what we see here or kind of along maritime Canada, where things like the polar cod are starting to have competition with Atlantic cod. So there's definitely going to be changes, but kind of trying to predict those consequences are obviously really complex. But another example of that is the terns with kind of eating Gulf of Maine fishes like herrings versus mid-Atlantic species like butterfish. That's obviously an extreme. Not all mid-Atlantic species are bad to eat. But just another example of how climate change is definitely going to affect how predators interact with their environments and what prey they're feeding.
Thank you so much. I had a question with that fish counting fence.
Yeah.
Did that create any bottleneck, or does it still as a fence for movement of the fish?
So I didn't pay Aaron to ask that. But some of Adam's other work, so we actually tagged the sockeye salmon smolts and looked at their movements. We used both acoustic telemetry as well as another technology I didn't discuss today, passive integrated transponder tags or pet tags, or same as microchips you put in dogs. But with the acoustic telemetry, I thought it was in the slides. Sorry, folks, it's very distracting. And this is some of that work. But yes, the fence seems to be affecting how well the smolts can get through this area. So for example, if we release a smolt just downstream of the counting fence, keep it going there. Man, I really thought I had these slides. I apologize. You can just tell I just keep adding more and more slides to the same talk. Here we go. Okay, so this is that same positioning system, but now you're seeing the path of one smolt that was tagged. This one makes it look super easy. This is 70 minutes. It goes to 1.3 kilometers, goes from the lake, which is the bottom of the screen, through the counting fence, which is that V at the top. But then we have fish that looked like this. And this is a fish. This is a fish that took 48 hours to go that 1.3 kilometers. Now, for context, usually within 48 hours of being released downstream of the fence, these fish can make it a couple hundred kilometers downstream. And usually they do the first 13 kilometers within the first night. And you might be thinking, some of you might be keen, like, well, Nathan, are you really tracking a smolt? Could you be tracking a bull trout then? Right? That's the first thing I thought when I looked at this. But one, it actually makes it through right at the end. And bull trout don't like going through that fence because it's so shallow. They make the water shallow so the fish can't stack on top of each other to count. 2, 6 days later, this fish was detected 600 kilometers further downstream, entering the Pacific Ocean. So this is a sockeye. And so Adam's doing a lot of work using what we call time to event analyses to see how long it takes smolts to get through this fence. And he found that things like fence operations. So, for example, if only half the fence is open, fish experience delay versus if the entire fence is open. Similarly, there seems to be a social aspect. If you're traveling with more friends, you're more likely to get through the fence more quickly. And that's some other work that someone wants to ask a question about predator swamping that we also looked at the system, kind of density, kind of survival of these migrations too. So we are working with DFO to talk about how this affects the population, but we should also note that this is the single best indicator of sockeye salmon juvenile production in British Columbia in a series of 70 years. So they make management decisions for about 100 different populations of sockeye salmon based upon data in part from those specs. So it's not necessarily something you can just rip out. In fact, in 2015, the year after these data, water levels were so high, it was unsafe to install the fence. Another climate change. They tried multiple other techniques, including acoustic imaging, as well as some artery capture studies that just did not work. And in fact, we tracked bull trout. It's another paper that Adam has in revision, right? And the bull trout still go to that spot. It's right. A fence is put there for a reason. It's shallow and it's narrow. But what we see is the bull trout stayed mobile a lot more and they moved a lot more in 2015 when there wasn't a fence to bottle everything up versus in 2014. But that would be super long. Question. Second back.
They have a question that relates to what you were working with. What kinds of collaboration discussions did y'all have? Was that related to the land? Was that related to the resources? Was it related to putting something inside a fish that they might be fishing?
Yes, kind of all of the above. I mean, it always starts with we have some interest in this. But what are your thoughts? And can we come with and there definitely were discussions of what is telemetry and what does it mean and what is going to be most vulnerable to the so it leads to things like protocols such that every bull trout that gets tagged also has an external tag, so it's very clear. And if a member of the indigenous community catches it, they get a very hefty reward. That's their foregoing dinner for that potential. And even some of the fish that we use were donated by them as well. Then it's often turns into what do you already know, and then what information would help you meet goals that you want. And so we're often in a very interesting position where we're working very closely with the federal government that doesn't necessarily have a lot of trust from them. But then also so trying to come up with research questions and protocols that work for both was often a lot of our efforts and time spent. Because we can only be there if they want us.
Well, I think I would like to, we've now hit the 9 o'clock hour, so I would like everyone to give Nathan another round of applause.
Thank you.
We were really lucky to have that range of information that was presented, all connecting a variety of different predator prey systems. And really, really glad to be evening. Next week is Tom Roberts from Brown University, and I do not remember what his topic is, so please look online and we'll find out. And anybody who's not here for another class next week, you can still join us online. Thanks again for your attention.